Today's topic is DNA, RNA and genetic code. We'll be looking at the basic concepts again in the form of MCQs. We'll be going into relevant topics which are possible for a medical entrance. We go to question number 16. Question number 16 says all DNA molecules have the same 4 nucleotides. You've got adenine, guanine, cytosine and thymine. Yet are different from each other functionally. When you take the functions, the DNA molecules are functionally they are very much different in prokaryotes as well as in eukaryotes. This difference is due to differences in the choices are A is the phosphate molecules, B is the sugar molecules, C is the sequence of bases and D is none of these. Now, as you understand, when you take the sugar and the phosphate in the DNA constitute the backbone. Hence, they are structurally similar in most of the DNAs. In all the DNAs, the sugar phosphate banister or the backbone remains the same. Hence, the variation, functional variation, like how do organisms differ in terms of that sort of a DNA, is the sequence of the bases, the stacking, the pattern of stacking of the nitrogen bases. Though the bonding is just similar, if you got the ATGC is the bonding, but the sequences of the bases is very much varied. Hence, the answer for question number 16 is answer is C, which relates to sequence of the bases. We say the sequence of the nitrogen bases. Now, question number 17 is a typical question which normally comes in an entrance exam, wherein the base sequences of the strand of DNA is cat, tag, cat, cat, GAC. If it is CAT, TAG, CAT, CAT and GAC, what is the complementary base sequence of the complementary RNA strand? What are the base sequence of the complementary RNA strand? Now, as you understand, when you look at the sort of sequences, the immediate way what a student should look out is if the first sequence is CAT. If the first sequence is CAT, naturally the alternative sequence should start with G. The alternative sequence should start with G. Hence, among the choices of A, B, C and D, A, B and C are near to this particular sort of a choice. But again, whenever you are taking A and U joined together, hence keeping this as a sort of a fundamental priority, the correct answer is D. The answer is D. So, always the student should note out what is the sort of an ATGC base pairing, and you should look out for similarities and differences, because given the sequence of the DNA, a sequence of the mRNA can be asked, or sometimes given the mRNA sequence, even the sequence of the DNA can be asked. Now we go to question number 18. In question number 18, it's asked the final structure of the double-stranded DNA coiled into a double helix forming spiraling major and minor grooves is dictated by what? It is dictated by the phosphodiester bonds which form between the backbone of the strands, that is choice number A. B is it dictated by the sugars which face one way in one strand, that is ring O up, and the opposite way ring O down in the other. And C is the base pairing between purines and pyrimidines, and D is the hydrogen bonds between purines and pyrimidine bases. Now we are understanding when you take the DNA structure forms the double helix, and when it forms the double helix, it goes to form the major and the minor grooves. The major and the minor grooves are the places where there is DNA protein interactions. We have DNA protein interactions. We have DNA protein interactions at the level of the major groove as well as at the level of the minor groove. Now, predominantly this is determined, like how a particular protein can interact with the DNA depends upon opposite polarity. It depends upon opposite polarity, whereby we understand there is one strand which is 3' has a 3' end and it moves to the 5' end, and we again understand there is a free hydroxyl group, OH group. Hence, if the sugars show opposite polarity, it facilitates the diameter to be equal and also facilitate correct sort of hydrogen bonding, and thereby even the DNA protein interactions. Hence, the answer to this is, the correct choice is B. B is the correct choice for question number 18. The double helix, question number 19, is a very simple question. It says the double helix DNA was described by Watson and Crick, who interpreted the results of, the choice A is Wilkins and Franklin, B choice is Michelson and Stoll, C choice is Hershey, and D is Holly. We know that Watson and Crick had proposed the DNA model. The DNA model was a deductive model, which was based on analytical studies. It was based on analytical studies as elucidated by the X-ray diffractions and as studied by Wilkins and Rosalind Franklin. Hence, for question 9, the DNA model is primarily deduced is based on X-ray diffraction studies. The X-ray diffraction studies could exactly know about the diameter of the DNA and of again the stacking of the nitrogen bases, and it conclusively also proved that there is always a complementary base pairing. Hence, the right choice is 19. The answer is A. Next, we go to question number 20. Replication of DNA in eukaryotes. In the case of eukaryotes, how is the DNA replication? It commences from, the choice A is from one end of the chromatid extending to the other, B from both ends of the chromatid simultaneously, C from the centromere to either ends of the chromatids, and D from several origin sites along the DNA of the chromatid simultaneously. Now, when you take DNA replication in eukaryotes, initially there is a formation of a replication fork. And on the replication fork we have the synthesis of the Okazaki fragments. And as the DNA fork moves, along this direction, we understand that the helicase enzymes unwind the sort of a DNA. It unwinds the sort of a DNA. So, parallelly, when you take DNA replication in eukaryotes, it is a method wherein you have two replication forks. Replication fork 1 as well as replication fork 2, and you have the presence of the helicase enzyme. So, the DNA replication occurs from both the ends of the chromatids, and it occurs simultaneously. Right from here as well as from here. Hence, for question number 20, the correct answer is, though it occurs simultaneously, when you take the entire sort of a euchromatid, such sort of a replication bubble, this is primarily termed as a replication bubble. This replication bubble has several origin sites on the chromatid. When you take the sort of a chromatid, if you think this is the chromatid, on the chromatid there are several origins. ORI1, ORI2, 3, 4, like that. There could be n number of origins. So when you take in the case of eukaryotes replication, this indicates the formation of 2 replication forks and formation of a sort of an extended area which is primarily an equivalent of the replication bubble of prokaryotes. Hence that for question number 20, the right answer is it has several origin sites. ORI sites are several. There are several origin sites and it occurs along the DNA and it occurs simultaneously. So presence of several origin sites and which occurs simultaneously is the most important thing, especially in the case of eukaryotes. Whereas when you take prokaryotes, in the case of the prokaryotes, you have only a replication bubble. It could have an ORI, single ORI, and this is called the replication bubble. So there's presence of a replication bubble and it goes in a particular pattern. While in the case of eukaryotes, there are several bubbles. Hence the answer to question number 20 is from several origin sites along the DNA of the chromatid simultaneously, and the answer is D. The answer is D. Now when you take DNA replication, this is quite a easy question. The choices for DNA replication is it A, continuous and conservative? 2, is it semi-discontinuous and semi-conservative? C is semi-discontinuous and semi-conservative, and D is conservative and semi-discontinuous. So when you take DNA replication, it is always discontinuous. Why do you call it as discontinuous? You call it as discontinuous because of the presence of Okazaki fragments. The smaller fragments of the DNA which are formed on the lagging strand. When you take you have a lead strand and then you have the lag strand. This is the lead strand and this is the lag strand. So when you talk about DNA replication, we require an RNA primer with joins, and later on you require a polymerase. Which will come and stitch all these pieces together. So hence initially it is discontinuous. Initially we understand it is discontinuous, and second point is it is semi-conservative. When you say semi-conservative, only we have one parental strand and another is the newly synthesized daughter strand always. So the evidences for semi-conservative replication with parental and daughter strands has been given by Michelson and Stahl. Hence we understand that it is both and semi-conservative, and the answer is B. The answer is B. When you take question number 22, question number 22 says, which one of the following sequences represents mRNA coded by DNA segment having the base pair GC, AT, GC, 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 CA, AT, CG and AT. So when you look at the particular pattern, we exactly understand if it's starting with G, the option should always be with relation to C or with relation to D. That is, it should either be a CT or a CU. So given the sort of an option, we always have the option as CUC, CUC, GC and G, GC and G, and UG, U. Hence, the answer is D. We need to check out the right sort of an answer, especially when you look at we either have to know the DNA sequences or the pairing sequences and look in terms of whether there is a sort of a correlation between ATGC base pairing. That's the way how you go about with relation to understanding all these particular sort of problems. When you take the 23 question asks the core of the nucleosome. When you take the nucleosome is predominantly made up of the chromatin material. It's made up of nucleoprotein material. And nucleosomes is responsible primarily for coiling and the arrangement of the DNA and the proteins in the chromosomes. So, any particular nucleosome is made up of H2A, then again it's made up of H2B, that is 2 components of A and 2 components of B, as well as even H3 and H4. Hence, the right answer is D. So, when you take a nucleosome, it is made up of 2 components of H2A, 2 components of H2B, then it is made up of 1 component which is made up of H3 and another component which is made up of H4. We have got 4 important components with relation to the sort of a nucleosome. Next, we go to question number 24. Question number 24 asks, which among the following ribosomal RNA is common to prokaryotic and eukaryotic ribosomes? When you take both prokaryotic as well as eukaryotic ribosomes, when you take them, they could either be based on the sort of a Swedberg's constant or based on their sedimentation coefficient, that is on the basis of their molecular weights. The ribosomal RNA can sediment. And when you take all the ribosomal RNA, we understand that the 5S RNA or the rRNA is common both in the case of prokaryotes as well as eukaryotes. Hence, the answer is B, 5S RNA. 5S RNA is very important here. Question number 25 looks at how 2 nucleosomes are joined. When you take 2 nucleosomes, a nucleosome resembles a box, and both the nucleosomes are joined by a linker DNA. This is the linker DNA, which later on coils around this. We have a linker DNA. It is joined both by the linker DNA as well as it is also joined with H3. Histone 3 and linker DNA are very important components. Hence, the answer is B. Answer is B. Question number 26 deals with very important discoveries, especially with relation to Nobel Prize in Physiology and Medicine. It asks, who synthesized nucleotides in vitro? In vitro refers to test tube, or it also refers to artificial synthesis. So, artificial synthesis of nucleotides, that is how do the nitrogen bases join together. This has been discovered by Mathai. Along with Nirenberg. and Hargobind Khorana. So this was the first evidence to prove that nucleotides could be artificially synthesized, hence it is the basis for cell-free protein synthesis. Cell-free protein synthesis. Hence the answer for 26 is, it's Mathai, Nirenberg and Khorana, hence the answer is D. 27 asks questions about the structure of the DNA. It says when you look at the structure of the DNA, which of the statements is wrong? So we are going to identify the wrong statement. A says DNA can be single-stranded in some virus. We have single-stranded DNA is present in viruses, which is a right statement. Then B says RNA can be double-stranded occasionally. Presence of double-stranded RNA has also been reported. C, there are as many as 12 base pairs per turn in the Z DNA. When you take the Z DNA, in the Z DNA, there can be as many as 12 base pairs allowed. Though there are 10 base pairs allowed, 12 base pairs are also allowed. Whereas when you take choice number T, when you take the choice number D, in D bit it says the length of one helix is 45 in B DNA and 34 in Z DNA. So it talks about length of the helix. Now length of the helix is always constant. Hence D is a wrong statement. Hence the correct answer is D. The odd is D. We go to question number 28. Which of the following statements is true for all tRNA? When you take transfer RNA, tRNA is also called soluble RNA. It's also termed as adapter RNA. Soluble RNA, it's also called as adapter RNA. So which of the following statements are true? The 5' end is phosphorylated. We have a 3' end. We have a 3' end. And again we have a 5' end. It asks whether the 5' end is phosphorylated. Yes, it has been phosphorylated. Then 2, there are single chains. Always when you take the RNA, it is predominantly single chained. It is single chained. 3, the anticodon loop is identical. When you take the anticodon loop, the anticodon loop of all the DNAs are not identical. And the 3' base sequence has a CCA sequence. This is a very important marker point for the tRNA. When you take the 3' end, it has got a CCA sequence, and the CCA sequence is the very important sequence in the case of the tRNA. Hence, the correct answers, it says in choice number C, it says statement number 1 that it is phosphorylated. 2, they are single chained. It's a single chain DNA, RNA I mean. And when you take 4, the 3' has got CCA. That is, you got and 4. So this is the right answer. Hence, the answer is C. Answer is C. We go to question number 29. Question number 29 says, nucleosome is an elliptical bead-like structure. And it has a bead-like structure whose length is 110. And it has a diameter of 65 to 75 angstrom and a diameter which can range from 65 to 75. It says its core consists of an octamer of histones, 8 histones, and its surface is surrounded by a superhelical strand of the DNA, which could amount to how many base pairs? So this is a direct question. Normally a helical strand round the sort of DNA of the nucleosome runs to 140 base pairs. So the correct answer is 140 base pairs. Hence the answer is B. Answer is B. So it's important that we exactly know how many base pairs are the sort of a how many base pairs are along the sort of DNA which connects two important sort of a nucleosomes. Question number 30. Fusion reaction of DNA is due to removal of RNA, is due to phosphoric acid, is due to phosphoric acid, carbohydrates and nitrogen bases, and is it because of aldehyde produced by acid hydrolysis? Whenever we take DNA and when we wash it, that is, we wash it in water, hydrolysis, and the acidic components come out. So it is predominantly, it takes a sort of a stain, hence DNA is fuelogen positive. So fuelogen positive is due to the presence of an aldehyde produced by acid hydrolysis, hence the answer is D. Answer is D. Now we are going to question number 31. Question number 31 asks, what is the contribution of leaven? Leaven is associated with Chargaff's rule, X-ray diffraction data of DNA, chemical analysis of Mister's nuclein, or DNA structural model. Leaven was a scientist who had primarily studied the chemical structure of Mister's nuclein. So, Mister isolated nuclein. Leaven studied what exactly are the nitrogen bases. What exactly are the nitrogen bases? Later on, Chargaff studied the X-ray diffraction studies, and based on all these things, these are the historical edifices, and based on all this, Watson and Crick proposed the DNA model. Hence, the correct analysis is leaven is associated with the chemical analysis of nuclein. The chemical analysis of nuclein, hence the answer is C. Answer is C. Question number 32 asks, what are Okazaki fragments? And Okazaki fragments give rise to lagging strand, leading strand, master strand, or the sense strand. Now, when you take the master strand, master strand is the original DNA, and if it has got the nitrogen bases which go to form the nucleotide, it is otherwise termed as a sense strand. During DNA replication, we have the replication fork, and we have the separation of both the strands. The first strand which gets formed is termed as the leading strand. The leading strand always synthesized from the 5' to the 3' direction in the direction of the replication fork. Whenever there is a helicase enzyme which tends to remove the sort of a torsion. So, while it is removing the torsion, we have the RNA primer. This RNA primer is synthesized by a primase enzyme. The primase enzyme takes the primer, and the primer loads at a site near to the leading strand and synthesizes small bits of the DNA, and these small bits of the DNA are termed as Okazaki fragments. So, Okazaki fragments are always associated with this strand is termed as the lagging strand. The strand which gets formed second, the strand which comes late, the strand which is again it is repaired by DNA polymerase and gives rise to the new daughter strand. Hence, for question number 32, the answer is A. Next, we go to question number 33. In question number 33 it says the tRNA, the transfer RNA, recognizes the enzyme amino-acyl synthetase due to the presence of what? Due to the presence of an amino acid binding site, or is it due to an anticodon site, or is it because of the DHU loop, or you got a CT loop. So normally here when the tRNA is there, the transfer RNA, the tRNA recognizes the enzyme amino-acyl synthetase. It recognizes, the recognition is primarily attributed to the DHU loop. The DHU loop is responsible for recognition. So it recognizes this, hence the answer is C. Hence the answer is C. We go to question number 34. It asks, DNA synthesis can be specifically measured by estimating the incorporation of radiolabeled uracil, adenine, thymine or deoxyribose sugar. When you are talking in terms of quantitative analysis of the sort of a DNA nitrogen basis, it is primarily due to radioactive adenine, radioadenine. Hence the answer is B. Hence the answer is B. Question number 35 asks, by X-ray diffraction studies, the diameter of the DNA helix was determined as 10, 20, 22 or 40 angstrom units. We understand there are 10 nitrogen bases, and within the 10 nitrogen bases, we exactly know the diameter which has been determined by Wilkins experiment indicates 20 angstrom units. 20 angstrom units of the DNA. Hence the answer is B. The answer is B. We are going to question number 36. A DNA chain has always a 5' phosphate end and a 3' hydroxyl end. It has got a 5' phosphate end and a 3' hydroxyl end. The starter engine is 3' OH and the guard or the last bogie is 5' phosphate end. Hence the answer is B. We go to question number 37. What exactly is the function of the DNA polymerase? The DNA polymerase is associated with choice A is proofreading and editing for DNA replication, B is for unwinding the DNA helix, C for union of Okazaki fragments, and D is none of the above. Now DNA polymerase primarily helps in proofreading. It removes errors, it removes any sort of an errors, it also removes the RNA primers, and it also edits. There is editing of the DNA so that correctly the first proofreading gene and also the first starter gene is correctly put in during DNA replication. So the prime function of DNA polymerase in DNA replication is, first one is it is proofreading and second one is it helps in editing. Hence the answer is A. Answer is A. Question number 38. Which of the following statements regarding a double helical molecule of DNA is true? Each strand is identical. We understand both the strands are different. The Watson and Crick strand are primarily different due to the presence of the nitrogen bases. B. Each strand can replicate itself. Each strand cannot replicate itself. Both the strands need to identify. Each strand need to act like a sort of a template, and then a daughter DNA gets formed. And C, the choice C says the nitrogen bases are perpendicular to the axis. When you take the nitrogen bases, if this is the axis, this is the way how the nitrogen bases align themselves. So definitely nitrogen bases are perpendicular to the sort of an axis. And D, all hydroxyl groups are involved in linkage. This is an untrue statement. Hence, the correct true statement here in this is, you got answer is C. Answer C, bases are perpendicular to the helical axis. We go to question number 39. DNA strands are anti-parallel. When you say anti-parallel, they have opposite polarity. When you say opposite polarity, one of the strands moves in this direction, another strand is synthesized in the opposite direction. So the DNA strands are anti-parallel because of A, hydrogen bonds. Hydrogen bonds are weak bonds, it's only used during DNA replication. B, disulfide bonds. Disulfide bonds are at present at the edges. And C is, it is anti-parallel due to phosphodiester bonds. Now phosphodiester bonds link up the sort of a sugar with the sort of a phosphorus. And phosphodiester bonds tend to link to either to the 3' or to the 5' tail. And D, the choice is none of these. So the correct choice here is phosphodiester bonds. We have the phosphodiester bonds, hence the answer is C. We go to question number 40. During DNA replication, one strand acts as a template for another. This shows its nature is semi-conservative. The answer is semi-conservative. It is semi-conservative in nature, hence the answer is B. Now, when you say semi-conservative, it means we have one parental strand. Imagine the DNA to be made up of a red ribbon and a blue ribbon. The red and the blue ribbon separate. The blue ribbon, a dark blue ribbon, synthesizes another daughter DNA, which is a light blue strand. And the red ribbon will synthesize another daughter DNA strand, which is light red in color. This pattern of inheritance, wherein always a blue ribbon and a red ribbon keep on going from one generation to another generation, is primarily termed as semi-conservative replication. So during semi-conservative replication, always one of the strand has got the parental properties. Hence the choice is B. 41 asks the similarity between DNA and RNA. Is both of them are made up of the same pyrimidines? No, they have different pyrimidines. Both of them have same thymidine bases? Again, no, thymidine bases are very much different. And both have the same sugars, whereas you take the RNA, it has got ribose sugar. If you take DNA, it has got deoxyribose sugar. But both of them are made up of nucleotide polymers. When you take both of them, they are made up of the building blocks of the nucleotides, and they go to form a large polymer. So both are examples of nucleotide polymers. And how do they differ? If you take the DNA, it is deoxyribose sugar, oxygen which is removed. And again, they primarily they differ in terms of the presence or absence of thymidine. So the correct answer is the nucleotide polymers. That is, the answer is D. Answer is D. Question number 42, it says the DNA molecule thymine is 20% than guanine. If thymine is 20%, then guanine need be. The correct answer is, it primarily refers to 30%. So 20 plus 30 will make 50, and the other side you get 50, hence it is 100%, hence the answer is B. Next is 43, question number 43. What is the similarity between RNA and DNA? The similarity is both of them have got purines. Both have purines. When you take both the RNA and the DNA, both of them have purines. Both have the ability to replicate. They have the ability to replicate. Both have the same structural basis. They have a pentose sugar. They have nitrogen bases. Then they are linked by means of phosphates. And again there is a presence of hydrogen bonds, weak hydrogen bonds. Hence the correct answer is D, which says it is all the above. 44 asks, nucleolus was discovered by, the answer is, it's a straight answer, answer is A, that is Fontana. Fontana is associated with discovery of the nucleus. Whereas when you take Sheridan, Sheridan is associated with discovery of the cell theory. Robert Brown, nucleus. Altman, primarily with relation to biochemistry. So the correct answer is A, that is Fontana. Nucleus has been discovered by Robert Brown, answer B. Has been discovered by Brown. Question 46 asks, RNA differs from DNA. How does the RNA differ from the DNA? In having uracil. Always RNA is associated with the presence of uracil. The presence of uracil, hence the answer is C. Answer is C. Next we go to 47 question. Nucleosome is, what is a nucleosome made up of? It's made up of only histone proteins. Choice A is only histone proteins. B is DNA wrapped around histones. C is RNA wrapped around histones. D, DNA wrapped around acidic proteins. The correct answer is DNA wrapped around histones. DNA, which is all round, it is bound along histones, wrapped around histones, is primarily termed as a nucleosome. When you take the fine structure of the chromosome, hence the right answer is B. We go to question number 48. Purines of the DNA are represented by adenine and guanine. Adenine and guanine, hence the answer is A. This is a direct statement. Question number 49 asks, the number of hydrogen bonds between adenine and thymine, AT. AT is always by 2 hydrogen bonds. So the number here is 2. Hence the answer is A. Answer is A. Question number 50. Unwinding of the DNA is done during DNA replication. How does the DNA strands unwind? Unwind is due to A, DNA polymerase. No, DNA polymerase is an enzyme which is responsible for the synthesis of the daughter DNA during DNA replication. B is exonuclease. Exonuclease is an outside nucleus which is added, or outside enzyme which is added. And when you take helicase, helicase is a DNA unwinding enzyme. So the correct answer is helicase. Helicase unwinds the DNA. So when it unwinds the DNA, it creates torsion. So helicase, when you say unwinding, it means unzipping. Unzipping of the DNA occurs due to the presence of the helicase. Hence the answer is C. While you take the D choice is ligase. Ligase is a joining enzyme, whereas exonuclease enables removal of any particular sort of a nitrogen base from the sort of a DNA. So the right answer is helicase and answer is C. Now we go to question number 51. In question number 51, it is asked, which of the following is not correct according to Chargaff's rule? According to Chargaff's rule. Now we understand from Chargaff's rule, always the ratio of A by T is 1. So choice number A is correct. B, the total of the purines is equal to the total of the pyrimidines. A plus T is equal to G plus C. And again, C by G is also unitarily 1. Hence the choice D is correct. Whereas choice C is wrong. Hence the answer is C. Now it is very important that whenever we are looking at Chargaff's rule of base equivalence, the three choices A by T is equal to 1, as well as C by G is equal to 1, and again A plus T is equal to G plus C. Now all these three are right. Hence, every student should exactly be knowing how Chargaff's rule can be applied in different modes. All the three are right. So in an objective bit, they could just be giving you any one of these particular things. So this is base. The Chargaff's rule is enunciates complementary of base pairs. The answer to question number 51 is C. We go to question number 52. Question 52 asks, ribose sugar differs from deoxyribose in having A is 2 extra oxygen atoms, B 1 extra oxygen atom, C no oxygen, D is hydroxyl group. We understand this is a question which shows deductive logic. We understand when you take DNA, DNA is ribose from which 1 oxygen is removed. So if 1 raw oxygen is removed, we understand that in the case of the ribose, ribose should have 1 extra oxygen molecule. Hence the answer is B. Question number 53. According to Watson and Crick, how many base pairs are there in each turn of the helix of the DNA? In each turn of the helix of the DNA, according to the Crick's model, we have 10 base pairs. Hence the answer is A. We have 10 base pairs. Next we come to question number 54. Question number 54 asks, which of the following is associated with the nucleus? When you take the tRNA is the transfer RNA or the soluble RNA which is present in the cytoplasm. Then DNA is primarily present as a chromatin inside the nucleus. Whereas when you take the ribosomal RNA as a precursor is present in the nucleolus and it is reflected in the formation of the ribosomes. And when you take mRNA, the mRNA is present in the nucleus and it is involved in transcription. Hence the right choice is 54C is rRNA. The answer is C. We go to question number 55. The distance between two strands of the DNA, or the diameter is 20 angstrom units. DNA diameter is 20 angstrom units. Hence the answer is A. Question number 56. One of the following combinations is wrong. Adenine A always joins with T. Purine always joins with pyrimidine. Cytosine always joins with guanine. Hence the adenine guanine combination is wrong. So all the three choices AT, purine, pyrimidine, cytosine and guanine is right, and the answer is D. The odd is the statement number D for question number 56. We go to question number 57. What is a nucleotide? A nucleotide, as you know, is predominantly made up of a sugar plus nitrogen base, and phosphoric acid. If you take a sugar and another sugar, they are linked by the phosphate bases, and each of these have got a nitrogen base. So sugar, phosphate and a nitrogen base together is termed as a nucleotide, which forms the basic building blocks of the DNA. Hence the answer is D. When you take the answer is D. Question number 58 asks, which base is not found in the DNA? In the DNA, we have got adenine, cytosine and guanine, and the nitrogen base which is not present in the DNA is uracil. The answer is here uracil is predominantly associated with the RNA. Hence, the right answer is C. Question 59. In replication, new DNA strand is formed or synthesized in which direction? During DNA replication, always the new DNA strands are synthesized from the 5' to the 3' direction. Always remember, the 3' direction is on the left hand side and the 5' direction is present on the right hand side. So, DNA synthesis always occurs from the 5' direction to the 3' direction, because we have the replication fork. The moment of it occurs on the direction of the replication fork. So, a new strand which is termed as the leading strand, and then we have the lagging strand, and always the synthesis occurs in this particular direction, and the direction is termed as 5' to 3' end. Hence, the answer is B. Question number 60 is again a straight question. If one strand of the DNA base sequence has CAT, GAC, TAG, what would be the base sequence in the other strand? So, it's given as DNA, strand 1 and strand 2. On one strand, it is given as CAT, GAC, and again it is TAG. So, it is asked what is the sequence on strand 2. On strand 2, it should definitely be GTA, CTG, and ATC. Hence, the answer is A. Answer is A. How many nucleotides are present in one turn of the double helix? In one turn of the double helix, we have 10 nucleotides. The answer is B. We have 10 nucleotides. Question 62 asks, the function of the nucleolus in the cell is. The nucleolus is not secretory. Option number A is wrong. Nucleolus is not involved in the synthesis of the DNA. B. But it is involved in the synthesis of the RNA and the ribosomes. If you take the nucleolus is involved in the synthesis of the ribosomes. Along with RNA. So, RNA and ribosomes and the nucleus are related to each other. Hence, the answer is C. Question number 63. The base sequence on DNA is given as ATG, CT, TAG. What will be the sequence of the mRNA? As implicated by several of the questions, wherever there is an A, you got the choices are UAC, GAA, UC. Hence, the answer is A. Answer is A. Question number 64. The acidic character of the DNA and RNA. Why are DNA and RNA acidic? Is it due to the presence of purine bases, pyrimidine bases, phosphoric groups, and sugars present? It is predominantly due to the presence of the phosphoric acid. The phosphoric group makes DNA and RNA acidic in nature. Hence, the right choice is C. Answer is C. Question number 65. The length of one full turn of the DNA is 34 angstrom units. 34 angstrom units. Hence, the answer is B. Answer is B. We go to question number 66. The type of sugar in the DNA is a triose, tetrose, pentose or a hexose sugar. It is always a 5 carbon sugar, hence it is termed as the pentose sugar. It is a 5 carbon sugar, pentose sugar, and the answer is C. Answer is C. Question number 67. The length of the DNA segment is 340 angstrom units. How many base pairs are present in it? We understand primarily because there are 340 angstrom units, the probable type of base pairs which could be present are 100. 340 divided by 100 is equal to 34. Hence the answer is A. Answer is A. The 68th question asks, what is a nucleoside? A nucleoside is a nitrogen base plus sugar. It does not have a phosphate. Whereas if phosphate is present, the sugar plus phosphate plus nitrogen base forms a nucleotide. So when you take a nucleoside, in a nucleoside, the most important feature is the phosphate is absent. The phosphate is absent. Hence the choice of base plus sugar, that is answer is A. We go to question number 69. If a purified sample of DNA is kept in water, then the solution naturally it becomes acidic. So obvious choice is A, that is answer is A. DNA is always acidic in nature. DNA is acidic in nature.